This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 550, recorded on May 31st, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello there, Vincent.
Are you at the University of Fort Lee?
I am. It's got a limited enrollment.
Or is it Fort Lee College?
It's Fort Lee Commuter College.
Community college.
That's right. Fort Lee Community Junior College.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. Good to have you on this side of the river, Dickson.
Hey.
He's on the same side as you. That's right.
True.
And from Western Massachusetts, Alan Dove.
Good to be here on the same side of the Hudson as you.
As me or Dickson and
as you, Vincent.
Wait a minute. Let me get my geography thing.
All of Massachusetts
yeah,
is east of the Hudson River.
Of course you are.
That's right, because the Hudson. Oh, yeah, because the states bend out.
It goes north all the way up to Canada.
And our final participant is also on the same side of the Hudson, Rich Condit from Austin, Texas.
Hi, everybody. And as a matter of fact, I'm the only one, however, on the western side of the Mississippi.
That's right.
This is all true. Hey, I just wanted to say something right now. Not one of us, in being introduced, said anything about the weather.
No, we didn't.
I was about to.
Dickson, you can't do anything about it, right?
You can't. But it's a beautiful day out there, actually.
So I'm looking at the map, and Hudson kind of bends a little bit, but the problem is the Northeast juts out into the Atlantic.
Right.
So that's why Massachusetts is there. Yep. And there's Springfield, the only city I see on my map. At a certain magnification, you see Boston and Springfield. Huh. And then if you zoom out further, then it's just Boston, of course.
Those are the two. Yeah, those are the two biggest cities in Massachusetts.
And then if you zoom in, then you start to get all kinds of stuff.
Yeah.
Yeah, Massachusetts. Today is the first sunny day of the week. Man, it has been weird here. We've had rolling thunderstorms. We had actually
cold. It was cold for a while.
We had a tornado watch. You must have had that, Brianne, too. Because
we had a tornado warning, actually.
Warning or whatever. Wow. And they told us to go in the basement.
Yep.
No kidding. We had flood watch
earlier this week.
So we went in the basement.
And I should clarify something. Kathy expressed concern in an email to me after a couple of episodes ago when I got a tornado warning on my phone while we were recording.
That's right. Yeah.
And she said, "You should have gone to your basement." I am aware of the seriousness of the situation. I actually had my my aviation map application open on my iPad, and I was watching the radar and watching my house thread its way through the thunderstorms. I said, okay, if that gets any closer, I'm going to bolt. So yeah, it was a little bit of a risk, but I was aware of the situation.
Have you ever had one?
A tornado?
Yeah.
We had one just like a mile to 2 miles from my house a few years ago that hit the city of Springfield and a couple of the outlying areas, did a huge amount of damage and took the tops off trees. Yeah. Goodness.
My first year at Notre Dame as a graduate student was around Easter of that year. And it was called it was Palm Sunday. I guess it was Palm Sunday tornadoes. There were 50 of them in a 20-mile square area. And we were right in the middle of it. The University of Notre Dame the campus was hit by 3 of them, and many, many people died. It hit a trailer camp of people, and they just died. And we had no idea of what was going on. We actually wanted to go out and see one, because you saw one in like The Wizard of Oz or something. And then we saw the rain going parallel to the ground and figured this is a bad thing. And we went back in our dorm, which was actually a married student's dorm. And toughed it out, and we could see the windows bowing in and out and all kinds of stuff. And I hope I never encounter one. It's really a terrible thing.
Yeah, many people had their houses wrecked.
Yeah.
Exactly.
Just this week. Yeah.
Mm-hmm.
Right. My son lives in Wichita. One of my sons lives in Wichita.
Which?
Bruce. He's an air traffic controller, actually.
That was a joke, but
Yeah.
Sorry. Which? Okay, fine. And not too far from him was this little town. I think it was Greenville or Greenberg, maybe Greenberg, and the entire town was flattened.
Wow.
And that's the one that Leonardo DiCaprio adopted, and we're bringing back with all kinds of modern contrivances to prevent that from happening. Not to prevent the tornadoes, of course, but prevent the houses from disappearing. What a tragedy that is for people living in an area like that, though, right?
Mm-hmm. Tornadoes are tough.
But
so are hurricanes. The thing is, the tornado comes up in no time, right? Boom.
No, you get some warning on that, though. At least you got a couple of days with a tornado.
No, that's what I say. With tornado, you don't have days.
Oh, right.
You have minutes.
You have minutes. That's correct.
Yep.
Well, we've certainly talked about the weather now.
Yes, we have.
If you like this chat and if you like the science that we do here on TWiV, consider supporting us. Microbe.tv/contribute. There are a number of ways you can help us out, including giving like $1 a month at Patreon or PayPal. Really would help us out. And if you are driving right now, which you probably are, you're doing something else, I know you're going to forget by the end. But I was thinking the other day on the podcast players should have a button that you could push.
Yeah.
And give $1, but they don't. Someday they will. I want to ask listeners if anyone has had a problem playing TWiV or any of the other podcasts on the webpage. I got an email from a listener who said that, if you go to the webpage, microbe.tv/TWiV, there's always a player on the page where you could play right there. Very few people do that. Now most of the playing is on mobile devices, but I have one listener who says it's been very slow lately, and I don't have that problem. So I'm wondering if anyone else has had that problem.
No, Vincent, I was on the last podcast. I stayed home and tied flies, of all things, right? Because it was raining. And I heard perfect. It was a perfect podcast and no glitches.
So you played it on the web page?
Yeah, I did. Sure, on my computer.
Mm-hmm.
And it played fine for me. So
right.
I think, I don't know what that problem is.
Yeah, I suspect it is a local thing, but
could be.
Just checking. We have a follow-up from Steph Langle, who is one of our co-hosts on Immune and has been on TWiV before. I hope everyone on TWiV is doing well. Firstly, I enjoyed episode 546 in the discussion of Hep D in birds and snakes and coronaviruses in pigs and bats. However, as you can imagine, I have a couple of comments from the episode. Dickson, I love you, but you were killing me in this episode. There are a few things that I want to help clear up. Let me pause here. Dickson did not know what it meant to say, you're killing me. I assume everyone else knows, right?
Well, I had an inkling. I knew it wasn't physical, but...
No. Steph is very nice. I don't know where it came from. It is a relatively recent thing.
Yeah.
Right? Here we go. Urban Dictionary. A phrase used when working with another and their performance is making you work harder. Oh, Frank, you're killing me. Lift up your side of the table. Slacker. But that's not how she meant it.
No, I don't think of it that way.
You're killing me, meaning you're saying things that are wrong, and I know it, but
exactly.
I can't respond. I can't respond.
Come on, I've got a reputation to maintain here.
Yes. All right, back to the letter. At 51 minutes, Dickson states that swine feces is more liquid than solid. That is not true. Normal pig feces is formed and solid. If they have an enteric infection, their stool becomes softer during subclinical infections and watery, mucoid, or bloody during clinical disease. And she sends some pictures for reference from the nationalhogfarmer.com. We have formed and normal. We have soft, wet, cement consistency. We have watery, mucoid, and bloody. That would be like this. That would be like
represent some kind of clinical disease happening.
That would be like dysentery, right, Dickson?
Yep.
Yeah, but yes.
And we have nice pictures of all
my ignorance.
Yeah. So pig feces is not.
Okay,
Well, okay, Dickson, you got that?
I got it.
Now, the thing is, Stephanie did her PhD on pigs, pig immunity, and she worked with a lot of them, and with 600-pound
those
sows.
And she has probably seen all different types of
Yeah,
pig poop.
certain.
She knows her pig poop.
That's exactly it.
There's the title.
There it is.
All right, here we go. Next point. Dickson referenced pig lagoons as his logic to why loose stool is normal for pigs. The reason he thinks that is because the lagoons from large farm operations look like a body of water, and she sends a link to a picture.
Right.
Hog operations that utilize lagoons for their manure management system will house animals on slated floors. The manure falls between the slates, and then a water flush system pushes the manure below the slates into a lagoon. If the animals are housed on concrete, then the manure can be scraped and hosed off into the lagoon. The lagoons are a combination of manure, urine, and water. They are not representative of what comes out of the animal.
Human sewage is also liquid. Is also handled.
No, that's true. Sure.
Right, so this is handling it in a similar fashion.
Yeah.
Sure.
At 1 hour 46 seconds, Dickson states that the majority of pigs raised in China are outdoors.
Well, that's old school.
That is no longer the case. So you were right a few years ago or whatever.
Yeah, you were right a while ago.
China has made major investments in the swine industry, as pork is their largest source of protein. And that's why that paper was a concern, that outbreak that we reported, because a lot of pigs died.
That's right.
While 1/3 pork is supplied from small farmers with less than 50 pigs per farmer, and that's 90% of pig farmers. 2/3 is supplied by large indoor operations like those in the U.S. They even have built hog hotel farms in China. I've never seen anything like that in the U.S., and she provides a link.
And that link is amazing.
Yeah.
So these are vertical farms.
This is vertical farms with a different sort of twist.
These I knew about, but they tried that in Holland, and the PETA people got behind the movement and refused to allow it, and they did not make it happen.
PETA
so China
does not,
is an exception.
as far as I know, have any kind of a presence in China. They would would be apoplectic.
No presence in China. They'll eat them.
This is a funny picture. The guy waiting at an elevator, and the pigs are waiting for the elevator.
Yes.
Yeah. Well. No.
That's so cute. I don't know. Are pigs the answer? Why don't they explore some other protein source?
Because as countries industrialize and people get money, they want to eat meat. And in Chinese cuisine, the primary meat is pork. And so it's better than beef environmentally because it converts more of its feed.
It's the other white meat.
It's the other white meat, right? It converts more of its feed into flesh than cows do. But a better source of protein, would be maybe supplement your diet with small amounts of meat, but eat more vegetables, especially like beans, which is a great protein source. Sure. But that just requires completely reworking everything about the food supply system. And certainly as Americans, we're in absolutely no position to criticize these choices.
We don't point fingers.
Eating absurd amounts of beef. So
yeah. But
they eat a lot of fish and chicken as well in China. So
sure.
And I'm wondering how much of the pigs that they raise are exported. I don't know the answer. Okay, Stephanie.
I don't either. But I think it's probably a significant amount, but I don't know. I don't know what the status of that is.
Because that's a cash crop for China, then.
Sure.
Right.
Continuing with Steph, also you all discussed that one of the control piglets in the study died to lack of colostrum intake and wondered why. Unlike humans, antibodies do not cross the placenta in pigs. It's too thick, 6 layers. Piglets are therefore born A-gamma globulinic and get all of their antibodies, IgG, IgA, and IgM, from their mother's colostrum. Without colostrum, they will usually die of infection.
That's fascinating.
Mm-hmm.
It is.
Yeah.
Mm-hmm.
Yeah.
I wonder what other animals have thick placentas that prevent antibodies from crossing.
Good question.
What is a pig, and what family of animals is a pig? Does anyone know?
They're not ungulates. They're
not.
Hey, they're mammals.
Yeah.
Yes.
There's a lot of animals related to pigs, so that might.
So
be
the family is Suidae.
Right, that's just going to be pigs.
So I wonder if all those suidae have a thick placenta.
Artiodactyla is there. Yeah. So they're more closely related to giraffes.
Giraffes, buffaloes, elk. Holy cow! They got
they are ungulates. Yeah, but they're with an even number of toes.
Lastly,
cloven hooves. That's the thing.
Lastly, there was discussion as to why SADS coronavirus may extinguish itself out after persisting for some time in the herd. During outbreaks like this, the herd veterinarian will work closely with the farmer/manager to implement strict quarantine and culling protocols. Additionally, infected sows that are kept around will develop immunity to the virus so that in her subsequent litter, those piglets will be better protected because the milk will contain antibodies against the virus. Milk IgA antibodies specifically play a major role in the protection of suckling piglets against enteric coronaviruses such as porcine epidemic disease virus. Shameless plug. She provides a link to her paper all about this from her graduate work. Ellen, all this paper is another example why veterinary virology is so important, not only to protect our food supply, but to improve animal welfare and prevent the zoonotic transmission of viruses. I hope this clears a few things up. P.S. for TWiV listeners, make sure to check out Immune.
Yes.
Microbe.tv/immune. Steph is a postdoc in Sally Permar's laboratory at Duke University.
Very good.
Hi, Steph. Hi, Sally.
All criticisms taken with a total humbleness on my part, and I appreciate the update of information.
You are accepting of criticism in public for sure.
Yeah, I have to. I make a lot of mistakes.
But you argue with me behind the scenes a lot.
But that's okay. That's okay.
Yeah, I guess so. All right, we have a snippet in a paper for you today. And the first is a Journal of Virology paper. Now, both papers today were suggested by others.
Not Vincent.
Not me. Most of the time I pick them just to save everybody work. It's also bureaucratically a lot harder to have everyone chime in. But today, both papers were picked by others. This first one by Rich Condit. Rich, do you want to talk about this little snippet with us?
Sure. So, full disclosure, the senior author, Dave Bloom, is a good friend of mine. As a matter of fact, I've known him since he was a graduate student with Dick Moyer, before I even moved to Florida. Then he went away and did a postdoc where he learned herpes virus.
Who did he do a postdoc with?
I'm trying to think. Jack Stevens, does that make sense? At UCLA?
Maybe, yeah.
And then was an assistant professor and maybe even associate professor at Arizona State. I think it was Jack Stevens. I think that's right. Who the individual who discovered the latency associated transcript LAT.
Well, you could go back to TWiV 84. He was on it and listen and find out. Gators go viral.
Right. And Dave, after Sue Moyer retired, Dave took her office, and so we were next door office neighbors for quite a while. And so, I've always had an interest in herpes and a close association via David and other people at the university. So recently I was back in Gainesville, and I had a talk with David, and he brought this work to my attention, and I thought it was really interesting, so I wanted to talk about it.
Most of us have close associations with at least a few herpes viruses.
True.
Yeah.
So the paper is by Terry Edwards and David Bloom, just two authors, and it's called Lund Human Mesencephalic Neuronal Cell Line, that's LUMES is the acronym, supports herpes simplex virus 1 latency in vitro. So I want to give a little review of herpes and say why this is interesting and important. Very briefly, herpes viruses are fairly large double-stranded DNA-containing viruses with an icosahedral capsid contained in an envelope. They come in many flavors, including HSV1, herpes simplex virus type 1, that causes cold sores, herpes simplex virus type 2 that causes genital sores, Epstein-Barr virus. Mostly we're familiar with that as causing mononucleosis, infectious mononucleosis, also associated with a few lymphomas like, what is it?
Burkitt's.
Burkitt's, thank you.
Right.
Cytomegalovirus. If you are infected with cytomegalovirus in utero, it's a major cause of birth defects. Kaposi's sarcoma herpes virus, HHV 6 and 7 that can cause roseola. So a wide variety of viruses, all having in common that they have a latent phase of infection. So they will infect individuals, cause an acute infection where there's replication and formation of new virus. But in the process of that, the herpes virus genome circularizes, finds its way to the cell nucleus, and can stays there in a quiescent state for the rest of the life of the host. Herpes is forever.
Yes.
And can occasionally then reactivate and cause symptoms. I should emphasize that in the majority of cases with all of these herpes viruses, the infections are subclinical. You get it, you don't even know you have it. You're harboring latent herpes virus genomes, you don't know you have it. The majority of the population is infected with these viruses. Only a minority show symptoms, and most of us are infected with several, most notably HSV1 and Epstein-Barr virus. I should say also
varicella.
I left out varicella. Yes, chickenpox and shingles. I left out that virus. Very good. Some of these establish their latent infections in neurons, and that includes HSV1 and HSV2 and varicella. Some of them establish latent infections in immune cells, and that includes Epstein-Barr virus and cytomegalovirus and Kaposi's sarcoma herpes virus. But they all have in common establishing latent infections. And so in order to properly manage these infections and deal with them in the long run, we need to understand how latency is established, how it's maintained, and how viruses become reactivated from latency. And these have been very difficult problems, primarily because there is a dearth of appropriate model systems or natural systems where these things can be studied. So human herpes simplex virus type 1, for example, there has been no real good cell culture model to study latency. There are animal models. There are rabbits and mice can be used. They're not perfect. They're hard to scale up. You can only do infections on a pretty small scale, so it's extremely difficult to look at the molecular details. The cell culture
you can only make
model,
a mouse so big.
Right? And the mouse model, you typically...
And you don't want to grow 1 million of them. Yeah.
In the mouse model, you typically infect the foot pad and
I bet in China you could.
You probably
could be.
Maybe.
At any rate, the cell culture models, there are some differentiable cells that have been looked at. There have been primary cells that have been looked at. A lot of these are very difficult cultures to establish, very difficult to scale up, and there are issues with the experimental systems themselves. Most notably, a lot of these, in order to maintain latency, you have to maintain the infection in the presence of an anti-herpes virus drug called acyclovir in order to suppress the acute infection. And so people question the relevance of systems like that. So that brings up the current paper, which is looking at a cell line called this Lund human mesencephalic cells, neuronal cell line. This cell line was established following all the appropriate guidelines in Sweden from human fetal tissue. that human fetal neuronal tissue, brain tissue, that was immortalized with a retrovirus expressing the MYC oncogene, which is a transcription factor that is important for growth control. And so if, and it's, the MYC oncogene is maintained in these cells on a regulatable promoter called a TET-off promoter. And so the cell cultures are maintained in the presence of tetracycline where the MYC oncogene is expressed, and the cells look fairly normal cells, and they can proliferate, replicate for many generations so that you can get a lot of them. That's the first most important thing. Then if you remove the tetracycline and add a couple of other chemicals, including a cell-derived neurotropic factor, glial cell-derived neurotropic factor, and cyclic AMP, that help differentiate these things into neuron-like cells, they will cease to divide and assume a conformation that looks like a neuron. They'll put out a bunch of dendrites and et cetera. So this is a terminal differentiation. It's a fairly homogeneous cell culture. And so this has been known for some time, but in the Bloom lab, I think this is the first real look at the utility of these cells as a model for establishing a latent infection. And so what they do in this paper is first show that in their hands they can reproduce the characteristics of the cell line by showing you, there are pictures here and appropriate immunofluorescence stains to show you what they look like when they're proliferating and what they look like when they're induced. And sure enough, they cease to divide and put out a bunch of dendrites and express appropriate neuronal markers. And then they infect them and see what happens. Now, in these infections, what they do is infect at a fairly high multiplicity, a multiplicity infection of 3, under which conditions all of the cells, theoretically, should be infected. And they do maintain the cultures in acyclovir for a period of 48 hours at the time of infection and for 2 days afterwards. And I've actually corresponded with Dave some and talked to him since, and he said they started doing that because when they first did the experiments, it looked like there was some damage to the cell cultures when they did the infection without acyclovir. And they're starting to question that and go back and see whether they can reproduce this whole thing with no acyclovir at all. But at any rate, they keep acyclovir in for, that's the anti-herpes viral drug, for 2 days, and then they take it out. And that makes this system unique in that they don't have to keep the drug in. And then they monitor for herpes genomes and herpes virus gene expression. And what they find is that they get a burst of of expression of viral genes that you would associate with a normal acute infection over the first say 5 days, 4 to 5 days, and then that gene expression decreases to a very low background level, but the number of genomes remains constant. And in a figure later in the paper, you can see that at least during the establishment of the infection, it looks to me like the majority of the cells harbor herpes virus genomes. And since that stays constant, I would have to assume that, though they don't have the data in this paper to show it, that later on, the majority of the cells in the culture are still harboring the genomes. So this looks exactly like what you would expect for establishment of a latent infection. A burst of acute infection activity that then subsides and goes to a background level, but the genomes are in most or all of the cells and maintained in apparently a quiescent state.
Right.
And then they go in and they add a common inducer of reactivation of herpes virus. This is wartmannin. That's rat poison, right?
No, that's warfarin.
That's warfarin. Okay, sorry. Dang. Okay. Wartmannin is an inhibitor of phosphoinositol kinase 3, which is a kinase that triggers a signaling cascade, which when triggered can cause reactivation of a herpes virus infection. And what they see when they do that is that they get a burst, again, of lytic gene expression and a burst of infectious virus out the other end. There's a really nice figure here where they look by RNA scope at both a latency-associated RNA called LAT. It's the most abundant transcript expressed during latency. That's why it's called latency-associated transcript. And also looking at a lytic-associated transcript, ICP4. And they have a time course.
Tell us what RNA scope is.
RNA scope is simply a hybridization method to look at very low copy numbers of RNAs in individual cells, and it's set up so that you can actually color them with different colors. So you can look at two RNAs, or even more probably, at the same time and distinguish them with different colors. It's very, very sensitive. And so they show here that right after infection, you can see ICP4, which is a lytic transcript, and a little bit of the LAT transcript. And then over time, by 6 days, ICP4, 6 and 13 days, ICP4 is pretty much gone. The LAT transcript, as you would expect, persists, which is exactly what you would expect. And then when you add Wart-Mannin, you get a burst of the ICP4 expression, and as I said, a burst of virus. And they have other immunofluorescence experiments that shows essentially the same thing, showing you turn on not only ICP4, but also ICP8, which is, ICP4, by the way, is a herpes early transcription factor, and ICP8 is a single-stranded DNA binding protein, and that comes on as well. So it looks like if you add an inducer, you induce a lytic infection. So from everything I can tell here, this looks like an absolutely ideal system for studying latency and the reactivation from latency in a herpes virus infection. So I'm really excited about this. I talked to David just yesterday or the day before in preparation for this, and he'd just gotten back from a meeting of herpes virologists.
Must have been a big meeting.
Yes. Though the herpes guys subdivide themselves because there are so many herpes virologists and so many herpes viruses. And I forget exactly the context of this meeting, but Bernie Reutzmann was there. He's like 90, okay, and still chugging along. At any rate, he said he gave a talk on this, and it was actually very well received, which is the herpes people don't necessarily receive all stuff well, okay? So that's a good sign. So I'm excited about this, and I'm interested to see where it goes. This provides the capability to understand better latency and reactivation from latency at a molecular level. Yes, right. Dickson.
So, Rich, is this a synchronous culture, or are these cells all dividing at different rates?
It's synchronous from the point of view that when you add, my understanding is that when you induce differentiation by turning off the MYC gene and adding these other things, that they all pretty synchronously cease to divide and differentiate into neurons. So it's a terminally differentiated culture.
So which cells develop latent infection and which cells are killed by lytic infection?
Okay, so in a normal situation, a very good question. In a normal situation with HSV type 1, the virus acquires entry into the host. It infects first epithelial cells. HSV1, recall, when symptomatic, calls cold sores. So it infects the mucous membranes in your mouth, the oral mucosa, and replicates there, and that's where the acute infection happens. And then it infects neurons and primarily establishes, as I understand it, latency in neurons. So the genome gets into neurons, travels up a process to the neuronal cell body, and establishes latency there, and can occasionally reactivate and then travel back down the neuron to and then infect the cell that's innervated. These are sensory neurons in the case of HSV type 1. Infect the epithelial cell that's innervated and undergo an acute infection. So most of the acute replication happens in epithelial cells, and neurons are primarily a site of latency. Now, how much acute replication might go on in neurons, I can't really address that, and I think that that's kind of an open question. One of the questions that is an open question is how much neuronal cell death happens during reactivation. And
exactly.
People debate that question.
Because you said there was a burst of replication and cell death, right?
No, I said there was a burst of replication. Okay.
So what happened?
And the cell. Well, David says now they don't. Actually, he says he can maintain these cultures latently infected for up to a month. But he says that when I think I recall correctly, that he said that when they're induced, they don't get a lot of cytopathic effect. And I don't know ultimately the fate of them, whether when you induce them, they die, or whether they sit there indefinitely. I don't, there's no information on that. I don't know that these are confluent monolayers in the same way as we're used to, where you could actually see plaque formation. I doubt it. It sounds like the plaque formation, usually you get a cytopathic effect. And my understanding is that upon induction, there's no really gross CPE in these cells.
Rich, when you get reactivation in a ganglion, do some viruses reestablish latency in brand new ganglia by reinfecting them, or do you have to go to the end of the sensory nerve to be able to reinfect
and do the whole circuit again? Yeah. I don't know the answer to that. I'll have David listen to this, and there are probably other herpes virologists out there who maybe can help us address some of these questions. Yeah. I would kind of assume, because you don't hear people talk about neurons infecting other neurons, or neuronal derived virus infecting other neurons. I would assume that this is sort of a cyclic thing, that you could reestablish or reinforce latency from the acutely infected epithelial tissue.
Because the reason is that it could be that you have enough latently infected neurons after your first exposure to last your lifetime, or they may be reinfected throughout your lifetime. I'd be curious to know which one it is. The other thing I thought would be interesting, and maybe they're doing this, there are plenty of herpes simplex mutants that are defective at establishing latency, right?
Absolutely.
Like, and this is ICP0 comes to mind, and so it would be interesting to put them in here and see if it's the same here as in all the other systems where that's been defined.
And I'm sure that that's on the agenda, because David has a significant part of his career has to do with studying latency, and he's got a collection of viruses that meet those criteria. Also,
I'd be surprised if that wasn't just on the agenda, but actually in the pipette as we speak.
An important part of this too is, what are the characteristics of reactivation? Is there a cascade of gene expression? What exactly triggers it? What's the cell response and etc.
All the stuff that's been so hard to study in vivo. Yeah,
exactly.
So I think it's great, but it's not in vivo, right? There are things in vivo that are not going to be here, like an immune response, which is important. But I think you can learn from both. If you remember back on number 430, we talked about an explant system for mice, a ganglion explant system, where you can infect, you can establish latency, and then it can be reactivated by a stress, and that happens when silenced histones, which are keeping the genome silent, are phosphorylated, and that partially reverses the silencing of the histone, gets gene expression produced, and so that's all really cool, and you can do that in an explant system. And I remember Rich, you saying that David was a little bit suspicious of that finding.
Yeah, I don't recall exactly that, but I'd have to look over the stuff specifically again. But I have always had trouble with the explant model myself, because of course you're ripping a ganglion out of its natural environment. I kind of assume, well, I don't know, that I kind of assume that part of the reason you see reactivation under those conditions is because you're removing it from immune suppression. Okay.
And you're also stressing the cells.
Yeah, stressing the cells big time.
In the explants, they had to do something to reactivate. But, it activates a neuronal stress pathway, and you get activation of neuronal kinases, which phosphorylate the histones and partially reverse the silencing. And I would love to know if that happens here. And this is,
yes,
Monin
exactly.
is a different way of reactivating, right. I think, but it would be.
And you probably have to try a number of different ways of doing the reactivation. And David is all set up to do the appropriate experiments with looking at histone methylation and acetylation patterns, and et cetera. But my guess is, I hope that a bunch of other labs jump in on this as well, and use this system and see what they can get out of it. I want to read. Go ahead.
Oh, I was just going to say, the other really nice thing here is that you can really look at sort of the human-specific factors in cells that are somewhat more physiologically relevant than a really transformed cell line. So maybe there's some factor that is playing an important role in mice or in rats, which you could see in vivo, but something different is doing that same job in humans. Here you're actually getting to look in human cells.
So David's summary to me in an email was that the main features of the cells that make them a good model for HSV and perhaps other neurotropic viruses are they're human neurons, they're easy to propagate because of the MYC gene is regulated by the TWiV system. They are scalable, and it's easy to get 10 to the 7th to 10 to the 8th cells. And unlike many other differentiatable neuron systems that they've examined, these cells are more terminally fated, so they rapidly differentiate and are homogeneous. They don't have mixed neuron and other non-neuronal cell types present. David says, but I always include the caveat that these cells don't replace the need to validate findings in animal models and human tissues where able, but they do allow us to do some biochemistry and molecular analysis that could not be done in vivo because of the scale. So it'll be interesting to see where this goes.
Yeah.
But you can see whether or not a cell that's activated to produce virus particles can also be the same cell that stays on as a latent infection. You can actually see whether that same cell is killed by the activation.
And I think that's a really good question. It'll be interesting to see what happens with that.
Sure.
All right. Thank you, Rich.
Sure.
Yeah.
Very cool.
I will also tell the listeners I found this paper to be pretty well written and pretty straightforward to it's the parts where I don't usually think about some of these things. I thought it was really easy to read.
One of the things I like about it is it's straight old classic virology, Yeah. It's the kind of thing you could give an advanced undergraduate or early graduate student class, and they'd really not have a whole lot of trouble working their way through it.
That was my exact thought when I was reading it.
Yep.
All right. Moving on to our main paper, which was suggested by Alan Dove.
Yeah.
Why did you like this paper, Alan?
Well, so this popped up in my, I have news feeds on a bunch of things, including the PNAS table of contents, which, every week it's just this gusher of like 100 papers that pop up. So mostly I'm skimming through titles, but anything that mentions anything viral, I'm always kind of drawn to, of course. So this is, it's a paper in PNAS. It's open access, by the way. And it's called Protect, Modify, Deprotect, a strategy for creating vaccines to elicit antibodies targeting a specific epitope. And the authors, again, two-author paper, Peyton Weidenbacher and Peter Kim, who are at Stanford University and the Chan Zuckerberg Hub, BioHub out in California. And vaccines targeting a specific epitope, one of the big problems in vaccine development these days, as we've talked about a number of times on TWiV, is a lot of the viruses that we want to vaccinate against generate robust antibody responses against the wrong epitope. And that's, of course, evolutionarily beneficial to the virus. So you get a flu virus infecting, and it produces a robust antibody response against the head of the hemagglutinin glycoprotein. And that responds to that virus, but that's also the most variable portion of the glycoprotein. So then a slightly different strain comes along the next year, and it has a slightly different set of antigens on its head, and the antibodies you built last year don't respond to this year's virus, and that's why you have to get a new flu vaccine every year. A huge focus in universal flu vaccine development has been trying to get a reliable way to induce antibodies against the more constant regions of the virus, especially the tail or the base of the hemagglutinin, which changes very little between strains. And in fact, you can find and you can produce and get antibodies very rarely that are against this base portion or the tail portion of hemagglutinin, and they provide immunity against multiple strains of flu. But most people don't end up generating those kinds of antibodies. We respond to it's like we pick the obvious antigen, and that's the one that's going to change. But the tail portion that's a little more hidden, our immune system's less likely to see. Stem, sorry.
Yeah.
We say that the stem is immunosubdominant.
Immunosubdominant. Yes, the immunodominant part is the head.
Yeah.
Yes. Your immune system basically goes for the big shiny thing.
Yes, exactly. So that's a good way to put it. So what these folks did, just the title kind of drew my attention. Then I looked at it. I said, "Wow, that's a really cool strategy." It's kind of like something that organic chemists have done for a very long time when they're trying to synthesize a molecule and they want to carry out a reaction, but they know, when we do this reaction, it's going to react with the wrong part of the molecule.
That's right.
So we protect that part of the molecule by sticking something else on it and then do the reaction, so only the part we want to target gets hit. And that's basically what's going on here. I mean, it's not, once you see what they do, it's not that complicated a concept. It's just really cool they got it to work.
Right.
So they took monoclonal. First of all, they did this with, they proved it would work with a hen egg white lysozyme, which is an antigen that's like used for all kinds of immunology experiments. It's been extensively studied. We want to do we want to induce an antibody response. What do we use? Hen egg white lysozyme. So this is a very, very well characterized, and it's got epitopes that are known to be immunodominant, and epitopes that are less dominant. And so this was a good model for it. And what they do is they take antibodies against one particular part of the protein and stick those on, and then they chemically modify the rest of the protein with various forms of polyethylene glycol, which is this kind of general purpose, very benign stuff. It's in a lot of products that you probably use. It's completely uninteresting to the immune system. So they basically put a cover over the protein, but it's got this antibody stuck to one portion of it, so the polyethylene glycol can't stick there. Then they take off the antibody. So now the only part that's exposed is the antigen that they want to target, and they find that they can get specific responses against just that targeted region that they left uncovered.
So they've kind of made the subdominant epitope more interesting for the immune system and more likely to be seen. By
making everything else less interesting.
Exactly.
They covered up the shiny part, to continue your analogy. So then they moved to hemagglutinin and did the same thing, basically. They've got a monoclonal antibody against one of the stem epitopes that people have looked at for universal flu vaccines, and then covered the rest of the hemagglutinin in polyethylene glycol and then took off the antibody, and they find they do a number of experiments to demonstrate that they haven't messed up the structure of the protein, and they've got, they're really accomplishing what they think they're accomplishing. But then the sort of the money shot is that they give this to guinea pigs, right?
Mm-hmm.
Guinea pigs, not mice. Yeah. So they deliver this protein to guinea pigs. The guinea pigs develop antibodies against it that are mostly against the stem portion of HA, and they verify that those antibodies from the guinea pigs will stick to a number of different strains of influenza, which is pretty cool. Now, this is still a very proof of concept kind of experiment. They're not challenging the guinea pigs with virus. They're not using a kind of a model that would demonstrate whether these are neutralizing antibodies. They're really just seeing if the chemistry of this works, and it does. And they end by discussing, kind of iterating on this concept and coming up with more refined versions of it that could ultimately lead to vaccines that would give you broadly neutralizing antibodies against a bunch of different flu viruses, or extending it to other viruses. HIV-1 is another good candidate for this, where again the dominant antibody response is not useful. So if you could, generate a more useful response against more conserved epitopes on HIV-1N, for example, you could get broadly neutralizing antibodies in a vaccine that way, maybe. But I just thought it was a really, really cool approach.
So I think that what's interesting also is the chemistry that they use here. They are basically attaching the PEG to lysines.
Yes.
Right?
Yep.
Right.
And they early on make this observation with the hen lysozyme that there are some epitopes that are still antigenic even after you add the PEG, and they call these antigenic holes.
Right.
This concept. And so you can modify them, and it may be that there's no lysines in this epitope, or they're not accessible or something like that. And in fact, for the HA experiment, they actually added lysines to make sure that these head epitopes, they added 9 lysines to the head of the HA to make sure that it would be properly covered with the PEG molecules. So I thought that was pretty interesting. And they also find that I think the addition of these, in part, just alone, these lysines changes somewhat the exposure of those epitopes.
Right.
The other nice thing was that when they did that experiment with HA and added the 9 lysines, they showed that they were actually able to kind of predict where those holes would be.
Yes.
Right.
And do something about them.
Yeah.
So if we wanted to use it for HIV or for something else, they show that the knowledge we have now could actually help us predict those holes.
Yeah.
So is it likely that PEG would be used if this eventually made its way into people? Would they use PEG? Or would that...
I don't know what the I mean different forms of PEG probably have different statuses in pharmaceutical approval, but I know it's kind of a generally used compound that's
it's in foods, right?
Yeah, it's in foods. I don't know. It would depend on what the regulatory context is probably and how much of it has been tested, but you could certainly find other substitutes for that or ways to modify it. The concept is what matters here, I think.
I can tell you from my own morbid experience that PEG is used as in preparation for a colonoscopy.
That's right. Right.
And one drinks copious amounts of this solution.
That's one way to do it.
With an astonishing result.
Yeah.
Okay.
They don't always.
You don't die.
There can be other.
Reference the letter at the beginning of the show.
There are other ways to do that. Also, you can do other things like saline balance and so forth.
Yeah.
But PEG is one of the old ones. Yeah.
So
It looks like. I was going to say it looks like it's used in gene therapy as well. They PEG coat the vectors.
Right. Yes.
Oh yeah. Right.
Yeah.
So it's not like if they were trying to get this through the FDA that you'd be starting from scratch.
No. No, right. And I'm sure that's one of the reasons they chose PEG is that the regulators are not going to say you're going to inject what into people.
Yeah.
So it's a pretty benign thing.
I'm just anticipating the vaccine deniers, reaction to putting PEG, right?
Yeah.
They already say we don't want aluminum, we don't want mercury, we don't want PEG. So I'm hoping that right,
and they're gonna home in on the ethylene glycol aspect of it and say they brought antifreeze in the vaccines.
Yeah. Yep.
I want to clarify something here because in case I missed it, in the guinea pig experiment, they didn't actually demonstrate directly that the antibodies that the guinea pigs were making reacted preferentially with the stem.
That's right.
They showed that they had a broader spectrum of reactivity. That is, could react with other subtypes.
That's right.
Whereas the regular vaccine did not. However, they got other experiments to support the notion that they have enhanced the antigen for reactivity with the stem epitope, because they show that one of the experiments, well, two experiments that I really like. One is when they show that the pegylated antigen disrupts reactivity with monoclonals that are specific for those pegylated domains without disturbing the others. And the other is they they enrich a yeast library. They got yeast that express a variety of different single chain antibodies on their surface for different parts of the influenza or for a different subtype of flu. And they present this antigen to them and use it to select out of this library of yeast cells whatever's going to be selected. And what's enriched for is antibodies to the stem domain.
Right.
So there's all of those experiments indicate that it should be working right. They got to have a closer look at these guinea pigs.
Yes.
So the stem domain, is that the protection epitope?
Well, it can be. It's broadly protective.
Is it?
Right. So the stem is more highly conserved than the head.
Yeah,
I understand. I totally understand.
But it can be protected. Antibodies against it will definitely protect. Yes.
Some of the broadly neutralizing antibodies are to the stem, right?
Yes, correct.
Yeah.
So do they know what the sequence is for those?
Of course.
Oh, yeah.
Of course.
So why can't you?
No, here's why you can't do that. Because they're conformational epitopes.
Right.
You can't just make a peptide.
Okay.
And that's the beauty of both the lysozyme and the HA epitopes are both conformational dependent, so you can't just make a peptide. And so this is kind of nice because you're presenting it in the normal environment, right?
Right.
Yep.
Rich, you said pegylation. That reminded me we can make pegylated interferon and give it to people.
Yes, right.
Oh, that's right. Okay, so that's been through the FDA.
Yeah,
that's right.
I felt like I was like, wait, I know this is used, but I can't think of the example.
It was pegylated that did it when he said that, yes.
So, Brianne, my feeling has always been that the reason that we don't normally make broadly neutralizing antibodies to the stem domain is just that it's not bright and shiny enough.
Yeah.
Is that right?
Yeah, the specific reasons behind immunodominance are not completely clear. And I know more about them in the case of T cells than in antibodies and B cells, but it has to do with things like the affinity of binding and the amount of the protein that is actually being made, and so the volume of antigen, I guess. So it can be all of those types of things, I guess.
And in some contexts, also accessibility.
Oh, of course, yes.
But I've always figured that even if your immune system doesn't normally do it, then using something like this, perhaps you can trick the immune system into doing this, and you will wind up with immunity to whatever it is that you want your target to be.
Exactly.
You can do that in some models.
In some models.
Yes. Sometimes when you take away the immunodominant epitope, the subdominant one will rise up. In other cases, it doesn't.
Okay.
So that's one strategy that is being done here at Mount Sinai, which is to immunize with an HA where the head is irrelevant. It's not from a human virus. And then you get a response to that head. But then you go back with a second HA, same stem and a different head. And now you get a primary against the stem. So you're kind of focusing.
Okay. So, yeah.
And you're kind of focusing it on the stem by putting irrelevant heads each time. So you don't get a memory against the same head, which would distract from the stem as well.
Interesting.
And you're confident that neutralizing antibodies, these are neutralizing antibodies.
Others have shown, yes, these are broadly neutralizing, BNAbs, right? That's what they call them, BN antibodies. They definitely neutralize. So this is accessible on the virus particle. There's no doubt about it. Brianne, can it also be that there aren't B cells or not enough of them with the right antibody on their surface that's recognizing this immunosubdominant sequence?
Yes, absolutely. Actually, one of the immunodominant epitopes people have found with HIV cross-reacts with a lipid that's in our cells, and so those B cells are deleted. And the reason why most people don't make those antibodies against that dominant epitope is because there are no B cells.
And of course, for HIV, these stem epitopes, the antibodies only happen after years of somatic hypermutation.
Yes,
right?
They have very, very long antigen binding regions and a lot of hypermutation.
Right. So for some reason, they're not initially present, only arise after this. And that, in fact, they say it would be interesting to use this approach to see if we could induce broadly neutralizing antibodies against the HIV stem without having to go through all of this hypermutation. I don't know if you will or not. It may be the same issue, right?
Right. Yeah. I don't know.
They just have to try.
You got to try.
Yeah. They just have to try it. Of course, what happens in a guinea pig may not be the same as in people anyway. So
it's very true.
Anyway, I think it's a very interesting approach. I hope that in a couple of weeks, I am actually going to a meeting in Baltimore sponsored by the NIH on universal flu vaccines, it's called.
Oh.
Ha.
And they're going to have different approaches, and I wonder if this one will be part of them, because there are others as well as that are being explored. And NIAID is really interested in making a universal flu vaccine for obvious reasons. So you don't have to get one every year. It would be nice. It would be really nice. So I should say that Peter Kim used to be at MIT. He was a structural virologist, worked on influenza, and then he went to Merck. For many years, he was head of research.
Wow.
Yeah.
And then he departed and is back in academia.
I think I met him when he was at Merck, actually. Yeah.
And so you can go back.
Yeah.
You could
if
go
you're
home.
Peter Kim.
If you're Peter Kim, yes.
That's right.
Now, this Chan Zuckerberg BioHub, this is money from Facebook, right?
Yeah.
Yep.
And what is a BioHub? Is it a physical place? Or it's just they're giving money and they call it a BioHub?
I think it's mainly a grant-giving organization. It's like the Gates Foundation.
And they give money to people in California only, right?
Is that their deal?
I think so.
Actually, my grad school roommate is working for them now in an administrative role handling their grants program, so I should probably know this. But yeah.
Well, I think it's wonderful that they're supporting biomedical research, and we need to have new infusions of money. Unfortunately, Facebook money is it's tainted.
I mean, you can say the same thing about the Gates Foundation. The money came from a horrible business model. So what do you do?
And, the Koch brothers fund a lot of science.
The Chan
the
Zuckerberg
Sackler Foundation does a lot of nutrition research.
Yeah.
I
sense a trend here.
Yes, the Chan Zuckerberg BioHub is a specific partnership between Chan Zuckerberg and Berkeley, UCSF, and Stanford.
Right. Okay.
I understand that this is not the only one, and even if you have a principal and don't take the money, somebody else is going to take it, right? And so that's an issue. But many people who have Koch positions are very unhappy with that name, because when they first got it, it was a little bit different, and now they're supporting evil things in this country. So you can turn your head or you can do something about it. I just think I don't care about Microsoft so much, because that's selling software, but Facebook is selling people's private information.
Well,
and lying about it. And
well, there's been a lot of backlash recently against the Sackler family.
Yeah.
What did they do?
Purdue Biopharma.
They supported a lot of.
They created the opioid epidemic.
Ah, right.
Yeah.
They had stock in a lot of those companies, and they made a fortune.
I think it's highly unfortunate that MIT has a Coke center, right? And
yep,
right.
That's great, but it's kind of dirty money, right?
We had a dean at our medical.
But it's money.
His name was Dean Merritt, back in the 50s and 60s, who was once quoted as having said, he was from Mississippi originally, so he knew a lot about backroom deals and stuff. He says the only thing wrong with tainted money is that it taint enough.
I understand. If we had sufficient NIH support, we wouldn't have to do this.
Exactly.
However, I will put it on the record, I will not take money from Chan Zuckerberg, from the Koch brothers, from Sackler.
Vincent,
and I will remain
go home and be very depressed.
That's okay. But that's just me. Don't worry. I just want to say that I can't do it on principle. All right. Cool paper. I hope it moves forward.
Yeah.
And just randomly, two author papers today. That's pretty cool.
Yeah.
Mm-hmm.
Right. All right. We have some email here. First one is from Anthony. He writes, "Good artists borrow, great artists steal," which is a quote of Pablo Picasso. And then in Jersey, anything's legal as long as you don't get caught, which apparently was said by Bob Dylan. And then he follows that with, you mentioned in one of the David Quammen episodes, using the title Viruses Are Us. Toys R Us used to aggressively protect their name. How that will proceed with the bankruptcy, I don't know. No matter. Use the viruses that are us, conflating Pogo. It struck me that you could start with perhaps a Liberty Science Center feature on the viruses that have found a place in the human genome. Maybe there could be a TWiV-tacular recorded there on the topic. If the topic resonates, a book could follow, and many more would learn of TWiV. Yeah, you'll see this. If the Liberty Science Center lets us do a TWiV there, never happen. Never happen. Because if I'm right, they want to do their own thing, which is what many of these big places say. Oh, we like what you're doing, but we want to do our own thing.
Right.
Rich, can you take the next one?
Johnny, our physician friend, pediatrician friend in Cambridge, writes, recall I visited her a few weeks ago. Did Rich share the pictures he took? I did, didn't I?
Yes, I remember seeing that.
I sent those around to everybody. Right. Not online, not in the episode, but I shared them with all the co-hosts. He photographed our state-of-the-art vaccine refrigerator, which was a hoot. It had a bunch of drawers with different diseases on the front of each little tray that had the vaccine for each disease. Maybe the incubator and sterilizer too, and a stadiometer where you can compare your height against some greats, Bald Eagle, Justice Ruth Bader Ginsburg, Dr. Seuss, Chewbacca, Emperor Penguin, and more. Yeah, that thing was great. Kathy referred to that in a subsequent episode. I appreciate and proselytize the work you and your distinguished co-hosts are so passionately committed to and make engaging, enjoyable, and educational, although more frequently I feel out of my depth with many of the laboratory methods, names of genes and their products, and the mechanisms of action. It's harder to visualize what's going on. Well, we're working on it. We're sensitive to that. But back to proselytizing, I tell every medical student and resident who spends time in our office about TWiV and the microbe TV. It is TMTV available for use. Lineup of shows. I also suggest the online lectures you and Brianne Parker post. Nothing like clear, well-organized lectures and teachers. Look forward to Sunday's podcast. It's like the Sunday Times arriving at the door. Something comforting, entertaining, and thought-provoking. Wishes of good health, strength, and warmer weather, Johnny in Boston. A little more than 10 degrees, overcast, and light showers. P.S. Check the April 2019 infectious diseases in children. And she gives a link to an article. Oh, I see. It's a whole issue on infectious diseases in children.
Yeah, cool.
Very, very cool. Yeah, we'll have a look at that. Got a bunch of different pathogens here.
So Johnny also posted a comment on TWiV 546, and I want to read excerpts. Sadly, much of the first paper on hepatitis D-like viruses went over my head. I was stopped and left stranded while the discussion went on without me. There were 2 stop codons. The first, that's a joke. The first was isoprenylation, an entirely new word for me. It conjured up chemistry classes and the thought I must have missed that chapter, lecture, and lab. I could not begin to imagine what it was and why it was important. Rich later gave a definition, but it didn't help me understand why or how this was significant to the discussion. Bah humbug. Then up popped the second stop codon, next generation sequencing. As my mother would say, I felt like a lost ball in high weeds. Maybe there's nothing to do but start a vocabulary list and look up what I don't know. It will cut into the flow of the podcast, but hopefully in future episodes, the weeds will become shorter and shorter and I won't feel as lost. So I'm sorry. Yes, we do throw away these terms and we will try to define them. That's why I asked Rich today, what is RNA scope, right?
Yes, exactly. Yeah.
And in general, and something ending in elation is we stuck this onto something.
Right.
So isoprenylation, we stuck an isoprene onto it.
Yeah, so this is a fatty acid that is stuck on the delta protein, and that enables it to be put into the capsid of hepatitis B virus. Right. Without it, it is capsidless. And there's nothing worse than a capsid-less virus, although those viruses would probably argue with me that they're very happy not to have it.
They're very happy.
Well, and it's sort of a similar word to pegylation, like we use today.
The isoprenylation is a baggage tag. Is pack this in the capsid.
So,
and next generation sequencing started as a marketing term and has just becomes kind of the standard thing that we use to refer to this. It means cheap DNA sequencing.
Yeah,
mostly
cheap and deep.
Cheap and deep. So because it's cheap, you can do a lot of it. So you just sequence the heck out of things.
So the PEG is interesting because I didn't remember until Rich said pegylation.
Pegylation, yes.
And that's a crappy word, right?
It is.
It's made up, but it's the addition of polyethylene glycol to various compounds. And in the case of the paper Alan talked about, it's to adding it to antibodies. And in the case of interferon, and Johnny, you might know this. You know, when you used to treat hep C with interferon, it was typically pegylated so that it would last longer in the bloodstream. Anyway, we'll try and be better about this.
And this kind of feedback is really good, especially the specific instances where we've lost people. Helps us understand where we need to improve.
I love the stop codon analogy.
Yeah, that's good.
That was great.
Unfortunately, I think we're riddled with stop codons.
Yeah, they're cryptic to us is the problem.
That's right.
Thanks for your persistence in the face of this.
Yes, we appreciate it.
We'll do the best we can.
We appreciate it. Alan, can you take Jerry's?
Yes, Jerry writes, here is the list of vaccine bills going around the country. There are 5 vaccine bills as of 2019 currently in debates, and he provides GovTrack links to these various bills in Congress. There's Senate Resolution 165, House of Representatives 1973, Senate 570, HR 1371, and HR 179. And these have different sponsors. Those are all on the federal level. And then he provides a series of bills in different states. Bunch of different states have vaccine-related bills. And then finally, here are all the states that have done their vaccine debates so far as of 2019, and another slew of bills. I feel like we ought to post some kind of. I don't know if we want to put this many links on the site, but people in these states should probably tune into some of these discussions and maybe pay attention.
So we've got Washington, Vermont, Texas, Oregon, New York, New Jersey. So these are all pro-vaccine bills.
Yeah, importantly, the important message here is that they're is a lot of pro-vaccine legislation being discussed.
Yes.
Yeah, I agree.
It's important to make sure that the pro-vaccine voices are heard.
Yeah.
Are heard. And the next letter is also from Jerry, right? Jerry?
Right.
Yeah, Dickson, you're a fan.
I'd love to read this.
Go
I'd love
ahead.
to read this one. Jerry writes, Dear TWiV, a Texas politician goes after Peter Hotez in the vaccine debates, and he gives a big link reference. I remember Peter Hotez from the TWiV special on Rachel. He got dragged again into the vaccine debates that is happening nationwide at a state capitol. Vaccine advocate Dr. Peter Hotez is accustomed to verbal attacks from anti-vaxxers, but on Tuesday, the abuse came from an unexpected source, a Texas legislator. In response to a Hotez tweet that the latest increase in vaccine exemptions in Texas shows its children have been placed in harm's way for the financial gain of special and outside interest groups, Representative Jonathan Strickland, Republican from Bedford, let loose. Quotes, "You are bought and paid for by the biggest..." I didn't do this right. I had to do this with a Texas accent.
You're bought and paid for by the biggest special interest in politics. Said,
well, thank you so much
Said
for the
Strickland,
backing me up on that. But actually, I think Rich could have done a better because he's down in Texas. Tweeted Strickland, "Do our state..." Go ahead, Alan.
"Do our state a favor and mind your own business. Parental rights mean more to us than your self-enriching science."
When Hotez replied that he doesn't take a dime from the vaccine industry, and that as a Texas pediatrician scientist who develops neglected disease vaccines for the world's poorest people, it is, quotes, "most certainly my business," Strickland dug in even deeper. Quotes,
"Make the case for your sorcery to consumers on your own dime," tweeted Strickland. "Like every other business, quit using the heavy hand of government to make your business profitable through mandates and immunity. It's disgusting." Well, immunity is actually what he wants.
Yeah.
he didn't even pay attention to what Hotez said. He doesn't get money. He's not getting paid.
This is a knee jerk. It's a knee jerk.
Yes.
Would we call
Is that what
Strickland
kind of
a knee
jerk
jerk?
he is?
You have a lot of people out there like that.
I mean, it's horrible that this person is a legislator, right?
It is horrible. It is horrible.
And-
But he's representing his constituency. And that's what you're looking at here. That is exactly what you're looking at here.
I want to point out, by doing that accent, I was in no way meaning to demean Southerners.
Notice I didn't do it.
No, and I'm from the South. I was raised in a Southern family, but I assume this particular legislator would have a voice kind of like that. And really, it's the words that matter. And calling vaccines sorcery is just hilariously stupid.
Correct.
I remember seeing these tweets when they happened, and I was kind of excited that this might have meant I was a sorceress.
Yeah. I saw this. I said, wow, I went to wizard school. Where's
Yeah,
my
exactly.
That's right.
This is very sad, unfortunately, because it's misinformation, right? The guy gets nothing. He gets no money, yet this guy is just saying it over and over, and people are going to listen to him.
Exactly right. He probably still thinks his vaccines cause autism.
So this is interesting. I'm looking at the article here, and it says after a certain point in the discussion we just had that Hotez sort of he bowed out of the tweet storm, but Strickland continued the onslaught with others happy to engage in the fight. In a span of an hour, he tweeted that vaccines are, quote, dangerous, that a doctor concerned about the child's vulnerability to disease is, quote, a brainwashed commie, and that a defender of science is a, quote, typical leftist trying to take credit for something only the Lord God Almighty is in control of.
Love it.
Wow.
Just so we understand who we're dealing with here, yeah.
Just use your voting rights appropriately, folks.
Yes, please. People of Bedford, yeah. I'm sure, I don't know who lives in Bedford, but I'm sure you can do better than this.
Should I say that they're standing in deep pig poo?
Maybe, yes.
So I got into a Twitter argument a couple of weeks ago, and with an anti-vaccine bunch of people, and, you cannot make any grounds. This lady said in the end, I don't believe a word you say. And
That's
then
right.
another woman came on and said, when these vaccines are tested not by the companies that make them and that have a financial interest, then we can talk. So they don't really. And this lady said, I'm not talking to you anymore. And I said, well, really, it was never a conversation, was it?
Right.
Bingo.
And I have since shut off my Twitter notifications because I am sick of such a cesspool of misinformation. I will go back at some point. But
That's right.
it is a cesspool for people who want to just spread misinformation.
Got it.
I mean, one lady said, after my child received this vaccine, he didn't speak for the rest of his life. And I said, this is the same trope that these individuals use over and over again. Something happened after a vaccination. And, that's the problem. We had a letter on immune this week, which was a wonderful, let me pull it up. It's a wonderful article explaining this business of when things happen together. Here we go. Letters read. Anyway, it's this whole idea that things happen, and when they happen when you're immunized, people think the vaccine is causing it. But unless you can provide an explanation for why this happened to the child, just saying it can't be the vaccine isn't good enough.
A week after my daughter got a whole bunch of shots in her thighs, she started walking for the first time. So in my experience, vaccines promote gross motor development.
Was the website the Spurious Correlations website?
No. It was an article in a letter from the Wall Street Journal a few days ago by Dr. Michael Siegel called The Complicated Truth About Vaccines. He writes, vaccines can appear to be the proximate cause of a condition when they have nothing to do with the ultimate cause. And the letter focuses on a 2010 study published in The Lancet that aimed to establish whether the apparent association of Dravet syndrome with vaccination was caused by recall bias, and if not, whether vaccination affected the onset or outcome of the disorder. And so they had an explanation for this Dravet syndrome, and they could show that it's not the vaccine. But unless we act like scientists and explain the science, we won't be effective in helping laymen put the frequently reported vaccination horror anecdotes in context. So I think that's a good point. We have to explain why things happen, not just say, it can't be this. Anyway.
Right. Yeah, I like to use the analogy about correlations, that there's a great correlation between the number of people who drown in a month and the amount of ice cream that's sold in a month.
Yes.
Yes.
And that doesn't mean that drowning causes ice cream, or that ice cream causes drowning.
Course. But people don't care. They don't want to hear it. They just want to know why their child didn't speak after being vaccinated.
Yeah, there's a wonderful website called The Skeptic that you should go to sometime and check it out, because this guy nails everything at that level. And, there's a lot of jokes about things like that, where a guy pulls the legs off a fly and then nothing happens, and he pulls the wings off the fly and then something happens. And I forget the tag into that, but it was totally opposite of whatever that experiment demonstrated.
Dickson, how do you name a virus isolated from a bat's fecal swab?
It's a badass virus.
Wow. That is from Dirk Jachmans in Belgium. Thank you, Dirk. And
Thank you, Dirk.
Brianne, can you take Lance's?
Sure. Lance writes, Dear Vincent and team, I don't know if I can really be called a listener anymore. I don't get time to listen nearly as much as I would like to these days. I am enjoying Immune very much. Here is a BBC radio program on science publishing that I'm sure you will find interesting, as will many TWiV listeners. Best wishes, Lance, and he is at the University of Liverpool.
And this falls right in our wheelhouse, the great science publishing scandal. Professor of Zoology, University of Manchester, explores the hidden world of prestige, profits, and piracy that lurks behind scientific journals. There you go. Okay, let's move on to some picks. I think that's enough letters for today.
Okay.
That's TWiV. Hey, by the way, we have TWiV 550. That's a nice number.
Yeah. Yeah.
That's a nice
That's
number.
a good one.
550. We've done 50 since our last year. It's a year, I guess, since last July's in Austin, Texas.
That's right. Austin, Texas.
Brianne, what do you have for us this week?
I have a website that I find really useful. It's called the FRED U.S. Measles Simulator. It's made by a group at the University of Pittsburgh. FRED stands for the Framework for Reconstructing Epidemiological Dynamics. And what this will show is 2 scenarios of a measles outbreak. One where fewer than 80% of children are vaccinated, so that's less than the threshold needed for herd immunity, and the other where 95% are vaccinated. And you can actually pick your state, and there are a few cities in different states, and watch that epidemic play out over 6 months.
That's cool. It's great.
Neat.
I show it to my students. The Essex County, New Jersey is the closest one to where we actually are in New Jersey. And that usually makes most of the points I need to make about vaccinations for me because the two are so dramatically different.
Wow.
Usually you'd think 80% was enough, but it shows how clearly that threshold makes
I'm just running Baltimore, Maryland right now, and it is dramatic.
Yeah, I'm running Manhattan, and you know what? There are no cases in Central Park.
How about that? No permanent cases.
Wow, a lot of cases would happen in New York City at a coverage of 80%. Wow, this is great. Yeah, next year I'm going to use this in my class.
I use it all the time with my students, and especially given the increasing numbers of measles that we're seeing right now, I think it's an important thing to show people.
Yeah, I actually use these numbers. I say you need 95% for measles, and this is a great way to illustrate it. Cool. Very nice.
Really good.
Alan, what do you have?
I have a place that I have just been going to recently. If you're anywhere in or near Western Massachusetts, or you can get here, you really ought to check this out on some nice summer day. Normally, it wouldn't be interesting to pick a reservoir, but this is the Quabbin Reservoir. It's the water source for most of the Massachusetts population. And it is an artificial lake, an absolutely immense body of water. It's the biggest lake in Massachusetts. It's one of the biggest lakes in New England. And when they created it back in the late 20s or early 30s, I think, they designated the whole area around it, which at that time was pretty empty anyway. They designated it all as wilderness to protect the water supply.
Right.
So the result is that today, within an hour's drive of my house and probably an hour and a half of Boston, is this enormous New England lake with nobody around. Wow. And the coolest part about it is that the story is the legislators who put this through and approved the construction of the reservoir. A lot of them were avid fishermen. And so they stipulated that the water department would have to allow boating, but could put restrictions on it. And so you can go to this place, and the result of this is you can go there, and for $40 you can rent a little outboard skiff. They also have a couple of places where you can rent a canoe or a kayak, but most of it, a couple of boat ramps. You rent a little skiff, and you go out on this huge lake, and it's like you're in Canada or northern Maine, and it's wonderful. It's a really, really cool place, and just absolutely worth checking out.
I'm surprised they allow outboard motors, though, Alan.
They had to. That was part of they had to allow fishing skiffs, which included that, and they have very stringent restrictions about horsepower limits and all this, and you can either rent one of the boats. You can actually launch a private boat, but it has to go through a decontamination procedure first.
Right. Got it.
Yeah. And there are some diehard Quabbin Reservoir aficionados, if you will, who do this whole thing, and they've got a boat dedicated to going to the Quabbin. And when they pull the boat out after a day of fishing, they get a seal put on, attaching it to the trailer so that it can't be launched anywhere else.
Right.
But do it the easy way. Go rent a skiff, get out there. You have to have a fishing license, which you buy for like $20, but you don't have to fish. So If you're not into fishing, you can just go out and float around on the water, read a book if you like, and it's freaking gorgeous. And diving loons and this kind of thing. It's really, really cool.
So you've been, I presume.
Oh yeah.
And when you go, do you see anybody else?
I'll see. So I rent a boat from the boat ramp, and of course there are people there, and you launch and you get away from the boat ramp, and off in the distance, you'll see another skiff somewhere, and at some point somebody might pass within a few hundred yards of you, but really sparse population.
It's
great.
Dickson, you've been there?
I passed it many, many times on the Massachusetts Turnpike because you can see it right from the road, but I have never actually experienced it.
Cool. All right. Well, sounds great. Probably never go.
Okay.
It sounds really nice. Rich Condit, what do you have?
So in 1992, my sister-in-law, that's my wife's younger sister, died at the age of 34 of a cervical carcinoma.
Oh.
And she was a rheumatologist at the Beth Israel Hospital at the time and had such a dramatic impact on her colleagues, in particular for her humanistic approach to medicine, that following her death, they created a foundation, the Catherine Swan Ginsburg Humanism in Medicine Foundation or program, that does its best to foster in doctors a compassionate or humanistic approach to medicine. And I'm bringing this up now because I just came back from the 27th. They every year they have a week of activities to highlight the program, although there are activities that go on all year long for fellows in the program. And they peer elect an intern and an attending to give an award for their characteristics that best embody the humanism in medicine ideals. And then there's a lecture by somebody who has an interest in this sort of thing. And the 27th meeting was this last week, so my wife and I went up there to attend this. I may have picked this before sometime, but I don't care. Every year I am so struck that this needs to even be an issue. Okay?
Compassion?
Yeah.
Yeah.
That it wouldn't be a stock element of being a doctor. At the same time, given all of the pressures involved in carrying out the discipline of medicine, it's not too surprising, I suppose, that some of the compassion and humanism that you might enter into the discipline with gets beat out of you over time. Yeah. Nevertheless, I just wanted to sort of highlight this program. This is a local thing that happens. It's associated with Harvard Medical School, as is the Beth Israel Deaconess Medical Center, where it happens. But just to sort of raise consciousness about the need for this and about the existence of programs like this that foster humanism in medicine. If you do a search, there's a search for humanism in medicine. You also come up with a national foundation called the Arnold P. Gold Foundation that has chapters all over the place that have I know less about this particular organization, but it has the same sorts of goals. And so I just want to, give a shout out to both of these organizations and hope that they continue on with this cause because it's really important.
Rich, Arnold Gold is a pediatrician at Columbia and suffers from scoliosis, unfortunately, and he has a heart as big and as generous as his last name, and has established the white coat ceremony for Columbia and for other medical schools too. I think it's now almost a national activity. The first-year medical students, when they first come to class, before they even go to a class, they're given their white coats, and they're treated after that as though they are young doctors in training. And he develops a series of programs which brings respect and humanism to the practice of medicine. And he's a generous person with his money as well as his attitude and his time spent with people. He's a marvelous person all the way around. So I'm so happy to see that Catherine Swan Ginsburg's Humanism in Medicine program is associated with this program as well.
It's cool. I think you did mention her previously, Rich.
I may have.
How long ago did she die?
92.
A long time ago.
So
wow.
the lectureship was started almost immediately. So it's 27 years ago.
Cool. All right. Dickson Despommier, what's in store with you?
Well, we have this new the James Webb Space Telescope under construction as we speak, and they've basically finished constructing it, but now they have to put it through its paces on Earth to make sure it can withstand the rigors of being stuck out about 1 million miles out from the Earth in naked space. So yesterday, I think it was yesterday, the day before that, it was subjected to the complete as complete a vacuum as you can get on Earth, with this entire telescope inside of this structure that actually evacuates almost every molecule in the atmosphere, and they lower the temperature, and they try to stress it as much as they can to make sure that when it gets out there, it can perform. But I look at this as a clinical trial. I mean, this is like great science. It's what they should have done to the space telescope before they sent it into outer space. They should have at least saw whether or not the lens was correctly ground or not. This doesn't have that kind of a system, so they won't have to check for that. But I just hope I'm around when this thing gets up and starts sending back data, because I think this is going to be this is going to be as earth-shattering to now as what the Hubble Space Telescope was when it was set up. This will go way beyond what the Hubble Space Telescope could ever have given it to us, and we're all attracted to that. So this has huge promise for just placing us in context with the rest of everything.
What's the timeline for this going up?
It's 2021.
2021, it says here.
2021,
yes.
Yeah. 2021.
So where is it going to be exactly 1 million miles
It's going
out?
to be 1 million miles away from Earth at the second Lagrange point.
Exactly. I would have said
orbiting the sun.
Oh, yeah, I know that.
It's orbiting.
Yeah, right.
So, okay, so it's in our solar system.
Yeah.
And where was the Hubble, or where is the
yes.
Hubble?
The
Hubble was orbiting the Earth.
200 miles above the Earth. And
okay.
It's in a decaying orbit, unfortunately.
Dixon, you want to go up with this telescope and stay there?
I don't want to go up with it, but I want to see what it has to show us. That's what I want to do.
So the Lagrange point, right, is where the pull of gravity of the Earth and the sun equalize. So
Correct.
it just sits there, right?
That's right. It's not going to move.
Neat. Cool stuff. How much is this costing, Dickson?
Lots, billions. Worth every penny.
But it was probably approved many years ago, right?
Oh, yeah.
Yes. But
In a kinder time for science.
That's right. That's exactly right.
My pick is a book by Erling Norby, who is a Swedish virologist. It is called Nobel Prizes in Life Sciences. Now, here's the story. In August, I have been invited by the Swedish Society for Virology.
Oh, my God. Oh, sorry.
No, that's a different time of year.
It's a different
To go to their meeting, which is on an island, Smogen, off Sweden somewhere, and to do a TWiV with Erling Norby about virology Nobel Prizes.
But the flight there is non-Smogen.
True.
So I've got 3 of his books. This is one of them. He's got 2 others about Nobel Prizes and so forth. And he's been involved with Nobel Prizes in various committees over the years. And so he's handled, and this is an interesting book, which talks about the prizes, how they were established. And there's one chapter here, the only Nobel Prize for a virus vaccine. Tell me what that is, folks. Yellow fever, Max Tyler.
Yellow fever.
It's the only one.
That's right. Does he talk about David Baltimore?
I don't know. I'm only halfway through, but he's got a chapter on polio and Nobels, unusual Nobels, Nobel Prizes in nucleic acids.
And if your data fails,
Nobel Prizes in prions. Anyway, so I'm reading this, and the other 2 to get ready for my interview with him, which should be fun. But this one is pretty cool. Nobel Prizes in Life Sciences, if you want a little insight into what goes on. As I said, he's been close to it. Check it out.
Very interesting.
Sounds good.
Then we have a listener pick from Eric. Hi, TWiV team. It's been a while since I've written in, but I have a couple of listener picks I'd like to pass along. I think the listeners will enjoy both, but one of them is entirely and shamefully self-serving. But that's fine. We do a lot of self-serving things.
Sure.
First pick is a new podcast that Boston's NPR news station WBUR is putting out called Infectious. It chronicles the history and present-day anti-vax movement. So far, I've only heard the first episode, but it was really good. It gave a history of the practice of variolation, followed by vaccination with cowpox, how the practices were received by a superstitious and alarmist public. The Infectious episodes fall under a podcast called Endless Thread. I've heard about that.
Mm-hmm.
The second pick is my art website that I recently launched. Like I said, self-serving, but I think the TWiV listeners will enjoy it nonetheless because my art is all virus-themed. I've loved painting images of viruses for years. I finally decided to put all of my paintings in one place, and the website is infectiousart.co. That's co, not com, because com was already taken. Let me know what you think. Keep up the spectacular work. Now, I have a work of art by Eric here in my office. He drew a painting of poliovirus. And Dickson, you have met him. He made us books once.
Yes.
He used to be into bookbinding, and he gave us 2 little books with a virus on the cover.
His paintings are amazing.
And
these are really cool.
These are very cool.
Yeah, he's got budding viruses.
He's got pleated sheets. He's got alpha helices. He's got.
I like the virus particles. They're very nice.
Does he do this professionally, like journal covers or something?
No, I think he just does this for. He's a scientist. He's worked in labs, and I believe he's going to graduate school in the fall. And
currently a PhD student at Drexel. Okay.
Yes, and he's. I have heard from him. We have heard for years from Eric. He has written to us many times. Born in Massachusetts. Yeah, so check it out, and you can buy his work to support his graduate career. So he's going to Drexel. Drexel, the home of
Philadelphia?
CD4 Hunter. Yeah.
Right.
All right, thank you, Eric. Thanks, everyone. This is TWiV. And if you'd like to find the links that we talk about, the letters, you can go to microbe.tv/TWiV. But most of you will listen on your phone. Some of you will listen on a tablet, and you use a podcast player, and you have the option of subscribing to TWiV. Please do so. It costs you $0. It is free, and it will always be free. And we ask in return that you enjoy it and learn, but if you really like what we do, please support us financially. You can give as little as $1 a month over at microbe.tv/contribute. We would be gratefully and eternally grateful. No, that's not right. We would be eternally grateful for your support. And of course, if you have questions, comments, picks, you can write anything. Just don't call us jerks. Which someone over on TWiP did last week. And, we don't do everything right, but when we get something wrong, just tell us, but don't call us jerks. Especially Dickson, because he's not a jerk.
Correct.
TWiV at microbe.tv. Nobody on this podcast is a jerk, actually. And, don't call us jerks.
No, to be a jerk, you have to not want to be corrected.
Exactly.
Right.
Yeah, don't call us jerks, but you can call us late to dinner if you want, but not jerks. Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
You're welcome.
Enjoy Fort Lee.
It's a beautiful little town.
Brianne Barker is over at Drew University on Twitter. She is BioProf Barker. Thank you, Brianne.
Thanks. It's been great to be here.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove's at turbidplaque.com. On Twitter, he's Alan Dove. Thanks, Alan.
Thank you. It's always a pleasure.
And I am Vincent Racaniello. You can find me at virology.ws. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.